Welcome back. We are starting with the chapter 17. This will be the last chapter for module 2, and we will be discussing the microbial DNA technologies, or sometimes we also call this as a recombinant DNA technology. The techniques that what is being used or in the market now is very sophisticated. We are not going to go into the details of that because some of them very minute modifications of these techniques can change a lot of different things. So we are not going to go into the detail of that. Rather, we are going to talk about just the overview of this recombinant DNA technology, or we can talk about microbial DNA technologies as well. So we're going to talk about the restriction enzymes, and we are going to look at the reverse transcriptase, cloning vector, what they are, and some of the different cloning vectors. And then we are going to talk about some of the key discoveries and relate to the development of the cloning technologies. And those are the things that we are going to talk about. So let's first look at some of the basic definitions that we are going to see. So genetic engineering is a modification of the genetic code of a living organism, including crop improvement and breeding in the animals. So what we exactly mean by changing a genetic code of an organism. And if you just look at the very beginning of your chapter in the book, just for your interest, that about a spider silk that has been modified by taking sequences from two different organisms, and there was a chimeric silk from the spider that has been made for many different purposes. That's genetic engineering. What is recombinant DNA technology? That recombinant DNA technology is when we take the DNA molecule with segments originating from different organism, we put it from a different organism, and we paste these. So say, for example, we are taking a DNA molecule from an eukaryote, we are putting that DNA molecule into bacteria, we are producing proteins out of that in the bacteria or in the yeast cells. These are the recombinant DNA technologies. So genetic engineering and recombinant DNA technology, that goes by hand in hand. You really cannot separate these two things as such. So on the other hand, what we mean by the DNA clone. So DNA cloning means that we do have a DNA segment of our interest. And usually most of the times it is not just a random sequence. The sequence is a gene of interest. So this gene produces a product, which is a protein, enzyme, or structural protein, and that is of our interest. So just for random, to think about that, we are talking about an insulin gene. This is an insulin gene of our interest. So now we just put that insulin gene into a vector which can produce this insulin product into a bacteria. This will be a recombinant DNA technology. Or sometimes what we can do, that we can take bovine insulin gene and then we can make a little bit of change in the codons, and part of that recombinant molecule is bovine as well as the human, and that may have some applications into the medical field. And this is a genetic engineering. So you can just correlate that these are some of the things or advancement of this technology. And biotechnology is a greater field altogether, which involve DNA cloning, recombinant DNA technology, genetic engineering, and in general that we use of organism to produce useful products. So say, for example, we use the vaccines, and the vaccine is now, you know, that coronavirus outbreak, it's really a cry for a vaccine. It's not there yet. But now, classically, say for an example, vaccines were made in different types of animals. And nowadays, what you can do just for the influenza virus, it is made in tobacco plants. So this product or the influenza virus antigens are made into the tobacco plants by recombinant DNA technology. So you just collect these recombinant tobacco plants, crush them, and isolate the protein for vaccine. Hepatitis virus vaccine is made in the yeast or in the E. coli. So these are some of the usage of biotechnology. So let's take a look at some of the key discoveries and which led to the development of the DNA technology. So in the 1960s, it was found there are some bacteria, or there are some archaea, and it was found that they produce some of these enzymes which can cut DNA in certain sequences, and why they do that. So sometimes there are many different reasons that these organisms actually produce these, and mainly for antagonizing different other threats, they want to chop the DNAs with these restriction enzymes. And that is mainly the purpose for those. And why we call this a restriction enzyme? Because these are endonucleases. That means that they will break the DNA or cut the DNA, double-stranded DNAs. But while doing so, they will do at a specific sequence, which is, again, we can think about that it is a restricted sequence, a specific sequence only. And this led to, in the 1960s and the 1970s, discovery of these enzymes. So these restriction enzymes completely opened up a new field where you can cut-paste the DNAs into a new organism. Now, what you mean by cut-paste DNA into a new organism? You isolate a plasmid from a bacteria, and you just cut this with a restriction enzyme. You cut a piece of genomic DNA from a mouse, and if the ends matches, and we're going to talk about that in detail, you can put that mouse gene into the bacterial vector, as in a plasmid, and that can be cloned into a bacteria. So we're going to talk about those. And genetic cloning and the cDNA is a complementary DNA that you can isolate the mRNA, and you can actually synthesize the gene of interest, because mRNA is the final message for the ribosome to make the translated protein. So mRNA, you have the full start to the stop codon, the full length of the gene without the introns. So if you can make a complementary DNA out of that, you get the full DNA sequence of the gene from start to stop. And then we're going to talk about the cloning vectors and what is gel electrophoresis. These are some of the areas that we're going to talk. So let's first talk about the restriction enzymes. So here what you are seeing is, on the right-hand side, just a molecular structure of a restriction enzyme, which is a dimeric molecule. It could be many different types, which is bound to the DNA. So the overview is that it recognizes and binds specific sequences in the DNA called the recognition sites. And these recognition sites are palindromic in the sequence. What we mean by the palindromic in the sequence? So when you read the sequence forward in your, again, I'm repeating, when you read the sequence on the 5' to 3' strand, the way it is read, if you read the sequence backwards in the 3' to 5' strand, then they are going to be the same. This is what is called a palindrome. So in the opposite strand, which is a sense and the antisense, or your coding strand and your template strand, the forward reading of the sequence and the reverse reading of the sequence in the opposite two strands are going to be exactly the same. So this is one of the main importance of that. These DNA sequences, where they find these restriction enzymes, when they find these sequences, they may cut them unevenly, which lead to this what we called as the sticky end. On the other hand, they can cut them blunt. The two strands are cut blunt, and we are going to talk about these as well. And then once we digest the DNA, we analyze these DNA fragments that we are cutting and pasting. Before pasting, we want to see that how they are, means that whether we have the correct molecular weight size or not, and that we're going to see by gel electrophoresis. We're going to look at that. And just a general idea that most of these bacteria, from where they originate, the restriction enzymes or the restriction endonucleases, that is the full name, but in short we call them restriction enzyme, named after the bacteria. So let's take a look at a restriction endonuclease in action. And this is a typical, we will going to see that this restriction enzyme is known as EcoR1. It is isolated from E. coli, and that's why it's called Eco restriction enzyme 1, maybe because it was discovered as a first restriction enzymes. So here I have a 5' to 3' DNA strand, 3', 5' complementary strand. And here we have a sequence, which is GAA-TTC. And here you can see that the complementary strands read backwards the same, GAA-TTC. So this is the palindrome. You read forward and you get the same thing in reverse if you read it backward here on these strands. So that's the key for any restriction site, that there are two complementary sides. You read forward and backward has to be palindromic. And the enzyme, for in this case, understand this sequence from both strands, and it's going to cut or nick the DNA in between the G and A here. So it will be G separated from AATTC, It's going to cut in between this G and A, so it's going to separate G and this AATTCs. And here is another site on the other side. And here in the middle, you have a DNA sequence. You have this, say, 1, 2, 3, 4 base pairs. This could be 4 to 400 or 4,000, doesn't matter. So here you have a sequence in the middle. Each end, you have an EcoR1 and an EcoR1. So if you take this piece of DNA and digest it with an EcoR1, you are going to get a fragment like this, where you have a GC and then TTAA, which is an overhang. Here you have a GC and you have a TTAA overhang on the 3' end. You have a overhang at the 3' end here, 5' to 3' on here. Here you have this 5' to 3' overhang AATTC. This middle piece, which also does overhang, and this is the piece that is cut out. Now think about that. We can cut out this piece, and then you can introduce this piece where the ends are cut out by the same way. This is going to ligate automatically. The sticky ends will go to ligate. We have to use the enzyme DNA ligase, of course, but these overhangs will base pair automatically, and all you need is a ligase to seal these gaps. So now, if your gene of interest is in the middle, having a restriction site at each end of it, and the vector where you are going to put your gene of interest having a restriction site, makes it easy to cut and then paste these DNA molecules. So that's the advantage of these restriction endonucleases. But again, here is one small thing which is a little bit extra from the book. Just remember that if we are, this is our molecule of interest. This is the gene that we are going to clone. At each end, we have this EcoR1 and EcoR1, which is the same restriction enzyme sites. Now, if we have this piece to be ligated in this, back again, this molecule can go in two different orientation. See that this AATT, instead of coming in here, this AATT can just go and bind in this region, and this AATT can go and bind in this direction. So what is happening? This molecule can be oriented in reverse orientation as well. And when it goes into the reverse orientation, there is a problem that when you are transcribing the mRNA, that sequence is going to compromise because your 5', 3', and 3', 5' end direction is now reversed. You will not have the proper product if it is needed to produce. Your DNA is cloned. That is absolutely fine. Doesn't matter whether it's forward or reverse direction. But if you have to use that particular gene to produce something, your mRNA or the RNA polymerase will not read and make the mRNA proper to its coding sequence, and you have a problem. So what we do that for all practical purposes, when we make these cuts, we cut with one restriction enzyme at one end, another restriction enzyme at one end. So the sticky ends that was going to be produced are always different in sequences. So this middle piece need to go in that sequence specific way. And there is no way that it can turn over and have this problem of this 3' end going to the 5' end and the 5' end going to the 3' end. It will always go in one direction because the sequences on this end and this end is different. And we're going to look at some of the examples soon later. So let's take a look at a few common restriction endonucleases. Say this is the one we were talking about, EcoR1 from E. coli, and it's GAATTC. And GAATTC this way, it cuts in between the G and A. So you can see that these are the overhangs that is being produced. So these are called the sticky ends because the overhang is going to be sticky and binding, making hydrogen bonds. It's very easy to seal these with the DNA ligase. On the other hand, this ALU1 is from the Arthrobacter luteus, and see that here you have an AGCT sequence, and this is a blunt end. So once this is cut, you will have an AGCT and you will have an AGCT. It is still palindromic. Remember that it is forward, backward is the same way. So that is the beauty of the restriction sites, that they are always in palindromes. And this is a blunted. Here it is a blunted. This is a overhang sticky end. This is a sticky end. So you can see that sometimes the restriction enzymes recognize, say here, 4 base pairs. Here it recognizes 1, 2, 3, 4, 5, 6 base pairs. Here is 1, 2, 3, 4, 5, 6, 7, 8 base pairs. So depending upon these, the restriction enzymes application, there are hundreds of them, restriction enzymes, just not a few, hundreds of them, almost every sequence you can probably find. But I'm just saying that they are going to be in all different combinations of these sequences. And the advantage of that is that some of them recognize just 4 base pairs, some 6, some 8. So depending upon these, the advantage of that, suppose you are trying to digest a whole genomic DNA. So the chances of getting these 4 base pairs in different areas of the genomic DNA is much more rather than getting these 8 base pairs. So this, if you are digesting a whole chromosome or whole genomic DNA of an organism, the chances of that DNA to be cut by this enzyme is more. It's going to make shorter fragments, whereas this is going to make larger fragments. And these are some of the applications that why they are larger or smaller. So let's take a look at the cloning technology and how this developed. And this was the first recombinant DNA technology or recombinant DNA molecule was made by the Jackson Simons and Paul Berg in 1972. And they generated the first recombinant DNA molecule, and the foreign DNA target can be recombined into a plasmid vector, and the plasmid vector replicate and maintain the foreign fragment, and you can produce a lot of these plasmids and purify them, and you have these clone of DNA into them. So how did we exactly do this? So suppose this is a plasmid vector, and you know this what a plasmid is. It's a double-stranded circular molecule, and this replicates in a permissive bacteria. Suppose you have an E. coli, and this E. coli replicates this plasmid, and all of these plasmid vectors that we are available in the market that we use, these are all engineered. They have very smartly produced areas or by recombinant DNA technology. Say, for example, just general, here you have a plasmid, which does have an AMP-R, or this is an ampicillin selection enzyme. On the other hand, what you have right here, you have a specific sequence, GAATTC, here palindromic GAATTC. Now you know that this area can be cut by an EcoR1 to produce 2 overhangs. You know it's going to cut here, cut here. So if you digest with EcoR1, now you have a linearized plasmid. Why linearized? This was completely circular. You cut in both strands, you have a linearized plasmid. So suppose this was a double-stranded DNA, completely circular, and this piece is out. And see that when it circularizes, a plasmid looks like this in electron micrograph. Now what you want to do with this? Now we have something which we call as a gene of interest. So this is our gene of interest. So right here, we have what we call as a donor DNA. So this donor DNA, suppose we are talking about a genome of any animal, like it could be yeast, it could be a mouse, it could be anything, it could be mosquito, it could be human being, and you want to clone a particular gene area into the plasmid. And suppose you have no idea that what you are going to clone, you digest the donor DNA with the EcoR1. Wherever this EcoR1 will find a GATTC sequence, it will cut those ends. So suppose you have a GATTC here, you have a GATTC here, you have this fragment, this is another fragment, and the gene sequence here and here is going to be different. They are just the only similarity that you have in between this fragment 1 and fragment 2, that each end of them has this overhang which is complementary to this EcoR1 overhang. But this is a gene 1, this is a gene 2. Now you take suppose the gene 1, each one of them have a sticky end, and you ligate it with the vector. And you can see that you have the CTA overhang right here. You have the complementary CTA overhang here. They will join, and you use the DNA ligase. You will have a recombinant DNA molecule. And this is basically the cloning process. Now what you do that once you have this, again, recircularized plasmid, This is a complete plasmid after the ligation reaction. You introduce this plasmid into the bacteria. Now, how do you put these into the bacteria? The way you put them in the bacteria is a process which is known as the transformation. If you remember, the transformation is taking up the naked DNA from the environment, and we can allow the bacteria to do that in the laboratory. And these bacteria are called competent bacteria. So a competent bacteria is a bacteria which it can take DNA or these plasmids from a test tube. So we add the DNA and the bacteria in a test tube, and we can do some specific process. There's a chemical process, and again, you do not have to go into the detail. We chill the cells for a long time with either calcium chloride or rubidium chloride, and then we heat shock these cells in the presence of the DNA at a 42 degrees Celsius for about 90 seconds, and it just takes up the DNA. There's another process of the DNA which helps in the transformation. This is known as electroporation. So we take these DNA and the bacterial cell in a small cuvette. It does have 2 electrodes in there. And in this cuvette with the electrodes, you pass a high voltage current for a very short period of time. So you pulse them with a very high voltage. So with that electrical pulse, it doesn't kill the bacteria, but with that small electrical pulse, the bacteria suddenly becomes competent and it will take up the DNA. The process is not very efficient, but it is enough to get some of the plasmid inside the bacterial cell. You just need just one bacteria to take up one plasmid. That's all you need. Then you can grow them out. So now what you do that you take these bacteria and you plate them onto an agar plate, and usually this is with a nutrient agar on which the bacteria will grow. So you have the bacterial food in there. But along with that, you have 2 other things in these plates. Not always, but there could be many different things. The plasmid do contain this ampicillin resistant gene. Ampicillin is in a specific concentration is in that agar. So the only bacteria, remember that I said that you mix the bacteria and the DNA and do the transformation. There will be many bacteria which will not be able to take up the DNA. If they don't take up the DNA, they will not grow in this plate because they will be susceptible to the ampicillin. Only the bacteria which does have this ampicillin gene in the plasmid means only the bacteria which has been transformed with the bacterial plasmid is inside them, they will be resistant to the ampicillin and they will grow. Now, here's also another thing that has been introduced. See that right here, that's been shown that this area contain a gene which is known as a LAC-Z gene right here. So what happens that when we introduce, so this is a continuous plasmid and you have a LAC-Z gene right here. And LAC-Z produce an enzyme called beta-galactosidase. And this vector, this plasmid, when there is nothing in here, in the growing in the cell, will produce this LAC-Z alpha and it will produce beta-galactosidase. But if I put a gene of interest right here with the flanking sequence of this overhang and ligate to that, and this is our recombinant molecule, that sequence of LAC-Z, which was continuous right here, is disrupted by that gene. So this plasmid will not produce the LAC-Z product, which is the beta-galactosidase. A plasmid without the recombinant piece of DNA or the DNA fragment will produce beta-galactosidase. A recombinant plasmid will not produce beta-galactosidase. Now here, there is a substrate, artificial substrate, which is inside, mixed with the agar as well. It's known as X-gal. So this X-gal is going to produce a blue color when it is hydrolyzed by the beta-galactosidase produced by the LAC-Z gene. So on the other hand, if you do not have any beta-galactosidase, there is no chromogenic reaction and all the colonies of a recombinant plasmid is going to be white. So what it tells us, the first, if we have any colony growing on this plate, that's going to be resistant to ampicillin. That means it does have the plasmid. Now, the second part, if the colony color is blue, that means that colony or that bacteria which is growing out of that colony does have an empty plasmid. There is nothing in there. It's producing lag Z. That's why you have blue in color. On the other hand, the white colonies, which is not chromogenic reaction, is not going on in here. You have the recombinant DNA molecule. You pick up those white colonies, grow them in a larger culture. You can grow litters of culture, and you isolate these bacteria. You break them up, and you isolate these plasmid by different techniques, and you have the cloned plasmid with your cloned DNA in there. And you can do a lot of different things using that. So this is basically your cloning technique and isolation of the plasmid, growing the plasmid, growing them in a recombinant cell. So let's take a look at what else can we do with this process. So the next thing, that how we can synthesize the cDNA and what it is and what is reverse transcriptase. So reverse transcriptase synthesizes double-stranded DNA from the RNA template or the mRNA, and used to construct the complementary DNA or the cDNA. This was independently discovered by Howard Temin at the University of Wisconsin-Madison, and David Baltimore was at MIT in 1970. They received Nobel Prize for that because this kind of revolutionized the way we do molecular biology and recombinant DNA technology, and they discovered it from a virus. It's a viral reverse transcriptase. That is the only place where you find that reverse transcriptase. And then those reverse transcriptase are now cloned. It's produced in large amount, and you can buy them commercially to do this process. So what is this cDNA synthesis? So for an example, if we are trying to clone a bacterial DNA from their genome on the chromosome, we do have a less problem, because the bacterial DNA, when they are transcribed, they do not have to process the mRNA because the bacterial genes do not have any introns. So you have an open reading frame with just only the exon. So you have a start codon, then you have a stop codon, you have the full length of the gene, there is no problem. But there is much problem with the eukaryotic DNA. Eukaryotic genes are broken up in pieces. So you have the exons, which code for the mRNA, for the real sequence that's in the protein, whereas the introns are the interspersed sequence which are needed to be removed by splicing so that exons joined to form a mature mRNA. So if we take the genomic DNA of the eukaryotes and try to clone a gene, we have a problem because of these middle pieces of these introns, and we just do not have the real gene. We can have the real gene, but with all of these intermediate introns. And that makes it a huge problem for cloning just the gene. Just the gene means from start codon to stop codon, if we need to produce a product out of that, which is the protein. On the other hand, what happens if we just isolate the mRNA or the matured mRNA from the eukaryotic cell, anything, or from an animal by grinding the tissues, we just isolate the mRNAs. It's easy to isolate. Now, what we do that if we have this reverse transcriptase, we can synthesize the complementary DNA strand. And this is the strategy that how we actually make the complementary DNA strand. Remember that your mRNA is 5' to 3', and at the 3' end of all eukaryotic mRNA, there is a poly A sequence. And these poly A sequences are almost in between 250 to 300 bases long. So there are 200 to 300 AAAs at the end of all the mRNAs. Now remember that these sequences outside its coding region, you have the start and then you have the stop, and this is after the stop codon. So now what you can do that you can artificially, this is done by the companies, you can just commercially buy these poly-DT primers. What is a poly-DT primer? Sometimes instead of poly-DT, it is also called oligo-DT. Oligo and poly means multiple, oligo is multiple, DT primers. So what are these? These are just a fragment of artificially made DNA with lots of T's. This could be 20 base, 15 base, 25 base, long TTTs. You have only thymines on this, and these are the DTs, deoxythymidine primer, only T's. Now what you can do that in a reaction mixture in a test tube, you have all your mRNAs that you have isolated from an eukaryotic cell. You add the primer which is an oligo DT or the poly DT primer. And these T's will find the poly A's, and they're going to bind by the hydrogen bonding because they are complementary. As they bind, now you have a primer. You add the reverse transcriptase. Reverse transcriptase is going to make a complementary DNA extending that oligo DT primer. So you had the 5' to 3' mRNA. Now you have a 5' to 3' complementary DNA, or 3' to 5' in the correct order DNA. So you have a single strand DNA hybridized with the mRNA. Then what you do that you add RNase H, and this RNase H is going to degrade this mRNA which was on the other strand. And there could be some fragments that's present in there. And at the same time, in the reaction mixture, you have this DNA polymerase 1 as well as DNA ligase. So what it's going to do, that DNA polymerase is going to take advantage of these residual primers, and they're going to extend these and fill all the gaps throughout. As the gaps are being filled, remember that all the RNAs are going to be eventually chewed up from the RNase H. So what is the advantage of the RNase H? Remember that RNase H only digests the RNA if there is a DNA-RNA hybrid. So that's very important. DNA-RNA hybrid RNAs are chewed up. And you can see that as they are being chewed up, as the DNA polymerase 1 is already in there, the DNA polymerase 1 used these templates for the primer and synthesize the complementary strand. Now, all these gap, there will be gap, of course, and these gaps will be sealed by the DNA ligase. That's how you get the 5', 3' strand back. So you have the 3', 5' complementary strand, you get back the 5' to 3' strand. So from the mRNA, you synthesize the complementary strand, and from there, you can get the full-length double-stranded cDNA or the complementary DNA, which is out of your mRNA. So this is the full length of the gene, but only the coding region, that means from the start to the stop, and some extra A's codon. So you do not have any introns in there, and that's why this is an extremely prized technique, because when you synthesize the cDNA, you are only synthesizing the coding region of the gene and you are eliminating the introns. That is the huge advantage of this. And now you can clone or plan to clone this DNA. So remember that you can introduce the different restriction sites at the end and you can clone this. And we're going to talk about that, how you can introduce those things. So now you have found a couple of different things. Let's take a look at it. Suppose you have a plasmid. Now you have cut that plasmid and you have an insert, which is this donor DNA. So now you want to analyze that plasmid and the donor DNA and want to know about their molecular weight. Or say in the next picture, you have synthesized this complementary DNA, cDNA from the mRNA. You want to know the size of it. To analyze these DNA fragments, what you do is a DNA gel electrophoresis. So how this DNA gel electrophoresis work? This is to separate the molecules based on their charge and molecular weight. So it could be agarose or acrylamide gels and can be used to separate the DNA. The most common is the agarose gel for most of the purposes. Acrylamide is used in a specific process in a vertical gel, whereas the agarose gel is used in a flat bed gel. And DNA is acidic, so it is negatively charged and it's going to move towards a positively charged electrode. And each fragment is going to migrate, and the migration rate is inversely proportional to the log of its molecular weight. So this is all confusing. So let's first describe that what we are looking at. So suppose we are doing a gel electrophoresis and we have a trough, just like a plastic trough. Each end of the trough does have an electrode, a negative electrode and a positive electrode that can be connected to a power supply to run electric current through that trough. Now, to run the electric current through the trough, that trough needed to be filled up with a buffer. So this is, just think about a glass baking tray. Instead of that, you have a acrylic baking tray, not a baking tray, similar, something like that. with 2 electrodes at each end, a minus and a plus electrode. And in the middle, you have an area where you have a particular holder of a gel. So what you do, that holder in the middle of the trough do have a square plastic like this, in which you cast a gel. How you cast a gel? You take the agarose, and this agarose is purified from a seaweed, and it is just like a powdery, and it's not going to mix with water. But if you just boil that agarose at 95 degrees Celsius, it melts. But it will solidify at about 47 degree or 45 degrees Celsius. So what you do, that you have in the trough in the middle, and you have a tray which is outside the trough without any water or anything. You have a tray which is sealed at both the ends temporarily. It's sealed at both the ends, and you have these two ends are closed all the time. So you have a square area where you can pour that gel, that molten gel, in here. And here you can use a Teflon comb to just hold it carefully on the rails. So you have a rail on this side, you have a rail on this side. So each side, right and left, the rails are permanent. On the other hand, the top and the bottom rails are just only used when you cast the gel and later removed. And you allow the comb to hang on top of the side rails so that they make some of these wells. Because once the gel is solidified, you can easily remove these Teflon combs, and they will have some specific holes in there, like slots, not really holes. This is where you load your DNA samples along with some specific dyes and some markers. So what's going to happen, that once the gel is solidified, you remove the gates from the two ends, and you put this whole tray in that trough that we talked about. And the trough is completely filled with a buffer. So on this top of this, there will be a buffer. Usually we use a tris acetate or a tris borate buffer, which will carry electricity. Now, you load your DNA samples in here, and usually it is mixed with a heavy material like a sucrose or phycol. So the DNA sample along with the dyes will sink into it. And then when you apply the electrical current, the DNAs are going to move towards the positive ends because they are negatively charged, because the phosphodiester bond is acidic and you have these phosphate groups. So it's going to move towards the plus end. Now, this agarose or the acrylamide, doesn't matter what it is, these are molecular sieves. That means that they will allow the migration of smaller molecular weights faster compared to the higher molecular weights. And the migration rate is inversely proportional to the log of its molecular weight. What do we mean by that? That the fragments which are going to migrate from this end towards this is going to be smaller in the molecular weight, which are staying towards the top are higher molecular weight. And how you can determine these DNAs? They can be added with fluorescent labels. They can be added with some dyes which fluoresce in presence of ultraviolet. Many of the different things can be added so that they bind to the DNA and you can determine where they are. Now here, what we are seeing, this is called a molecular weight ladder. These are commercially available from the vendors, and you can buy these. And they are also stained or fluorescent. And when you observe them, you can see that there is a ladder. This is 100 base pair, 200, 300 base pair. This is right here, say 1 kb or 1000 base pair. Here is 10,000 base pair or 10 kb. So it forms a nice ladder. So this is your reference lane. This is your molecular weight lane or reference lane. And on this lane, you run your DNA samples, vector, insert, all the different things. And from these lanes, you can determine the molecular weight of your sample, comparing it with the reference sample. So here, this is say in between 100, 200, maybe 150. This is just slightly over 200. This one is close to in between 650 and around 650, in between 700, 800. This is close to 600. On the other hand, if you look at that, this is probably in between 850 maybe. So you can easily determine the approximate molecular weight, not exact, but approximately molecular weight of that, so that you know that what you are doing or you are working with your samples they are of the correct size. So this is your gel electrophoresis, essential at every step. So now we are going to talk about the cloning vectors. We already talked about the plasmid. That's a kind of a cloning vector where you can insert the gene of your interest. But along with that, we have bacteriophages. We use viruses, cosmids, artificial chromosomes, and the DNA elements, they replicate and carry these additional cargoes, separate them in different classes. That's what we want to use. So here we are looking at a plasmid, a POC19. It's about 2.68 kilobase pair, 2686 base pair long. And this is a bacterial plasmid. And let's take a look at what you have. So all of them will have an origin of replication, which is the ORI. You have a selectable marker. You can see that AMP-R, this is the ampicillin resistant. That's the marker. Remember that we have a LAC-Z site, which makes that beta-galactosidase. You have that. And inside that gene, you have this you have this multiple cloning site starting with EcoR1. Now, what are these? You can see that EcoR1, SAC1, KPN1, and all these are the names of different enzymes or restriction enzymes which have their corresponding sequence right here. And what is the reason for having this multiple cloning site? It gives all the researchers a choice that you can use a combination of any of these enzymes to cut your sample DNA, and you can cut the vector with the same restriction enzymes, and they will produce the same sticky end, and they will be able to join those. Just giving an example. Say for an example, you just want to use just one restriction enzyme. Okay, fine. You just use EcoR1. So all your DNA fragments will have EcoR1 end at one end and EcoR1 at the other end. And you digest the vector with the EcoR1, and you can ligate your fragments with the vector, and you ligate them with the DNA ligase. Problem with just using one, they can go in a different orientation. But suppose you digest your samples with EcoR1 and say BAM-H1, EcoR1 and BAM-H1. So what you will have, you are digesting the vector with EcoR1 and BAM-H1 as well. So the vector will have a sticky end of EcoR1 at one end, sticky end of BAM-H1 at the other end. Both are different. So now you have digested your sample with EcoR1 and BAM-H1 as well. So only the fragments which has EcoR1 at one end and BAM-H1 at the other end will go into that multiple cloning site. If they're not double digested with two restriction enzyme, they cannot be inserted because they will not be complementary. So this is the advantage of this multiple cloning site, give a lot of flexibility to the researchers. Now, here is another plasmid, YEP24. This is a plasmid from the yeast. Yeast is an eukaryote, but that is the only known eukaryotic that actually produce plasmid, just like the bacteria. So here, there are a couple of different things. You have an origin of replication, which is also ORI. Then you have a selectable marker. Here is actually a couple of them. You have an ampicillin resistance. You have a tetracycline resistance. Then on the other hand, you have an URA3. This is a selection for the uracil auxotroph. You have all these different features in there. So you can easily use these different things. So you can put 10, 15 kilobase pairs or 10,000 base pair or 15,000 base pairs of DNA into these. But if your DNA size increases, you have to use something else. So the phage vector genomes are genetically modified, which can also include restriction enzyme recognition sites. A very popular one is an E. coli lambda phage. So you have the genome from the E. coli lambda phage, and the scientists have manipulated the genome of the bacteriophage and put this restriction sites on the multiple cloning sites. So what you do that you can just put that your gene of interest into the multiple cloning site. That particular DNA will be packaged into the virions of the bacteriophage. And now the bacteriophage can transduce, or they will just go and infect new fresh E. coli. These E. coli will grow this bacteriophage and they will lyse the cells and come out. Basically what you do that once you package and make this bacteriophage, which is a recombinant phage, think about that, what you're trying to do. You are taking these small few phage and you're taking a large amount of E. coli, suppose it is going in a kind of a suspension culture in a 1 liter flask, and you just add a few microliters of your bacteriophage or recombinant bacteriophage. It's going to go into the media and immediately attack all the E. coli's, and they are going to, all of them are going to now replicate that phage vector along with your gene of interest. So think about that, how much is going to be made, because this whole liters of E. coli is full of these phage vector DNA along with your DNA. So you just lyse the cells, you just isolate these bacteriophage vectors, and you will have large amount of your clone DNA which are in these phage vectors. So you just have a large amount to work with. So what happens is that, again, this is the thing, after insertion of the foreign DNA into the recombinant phage genome, is packaged into viral capsids and used to infect the host cells, which are E. coli, and there are many other different types, but the most common one is the lambda phage of the E. coli. On the other hand, genetic engineering of animal cells is accomplished with the viral vector. There are other processes as well, where you use some of these retroviruses, lentiviruses are retroviruses as well, and adenoviruses, where they are some of the essential genes of these viruses to make it a productive infection has been removed. So these viruses can go inside the cell and deliver the gene or the cargo, what they are carrying, but they do not have the ability to produce the lethal or the cytopathic effect so that they are going to kill the cell. They are not going to kill the cell, but they are going to deliver the gene of interest. So in this process, what you do, that you have the viral vector, the DNA, and you have a multiple cloning site where you put the gene of your interest and then you package them into the virion, and this can be easily done. And now you have the virus in the test tube with the vector inside it, and now you infect some animal cells in the tissue culture, and these viral vectors along with the virus will go into and infect all these animal cells. All these DNAs which are inside those vectors are going to replicate it into those eukaryotic cells. So you can isolate those cells and then break open them. You isolate the DNA from the viral DNA from there. All these viral DNA will have your clone. So what is the advantage? Why we are putting this in these phage vectors or the viruses? The main function is that to make a large amount of your cloned DNA. How to get the large amount? Once you put them in a vector, they are going to go in either in the bacteria or in the animal cell to produce a lot of copies of them. That is the cloning process, that you have a particular vector that is making thousands and million clones of itself. So you have a large amount of these recombinant product or the gene of interest that you want to work with. And what you want to do with that will be the next part. We will going to talk about that. Next, we are going to take a look at the next kind of vector, which are the cosmids. They do not exist in the nature, and you can see that as the name suggests that they do have something to do with a plasmid, and the cos is actually a site which comes from the cohesive ends. And if you have a cos site, these can be packaged into a bacteriophage. So here, this is an artificial vector where that vector has the ability to be packaged into a bacteriophage. Also, it does have a selectable marker, origin of replication, multiple cloning site. So this can be replicated into a bacteria as well. So this replicate as plasmid within the host cell, but in the presence of the cos site means that vector can be packaged into the phage capsid and transferred to a new host cell by transduction. So what is the advantage? Why we are trying to do that? So suppose you have a plasmid, but you have a insert or a sequence that is 25-30 kb long. You cannot put that in a plasmid because when you transform the cells by that process, it is not going to take up the plasmid, because the plasmid is so large. The transformation efficiency, or the competent cells that take up that plasmid, is going to be very, very less. So you're not going to get efficient transformation. It's almost impossible to get that big of a DNA into the cell by transformation. So what you do, you have this vector, which is a mixture of a plasmid with a COS site, and you have a multiple cloning site, and you clone your gene of your interest into that multiple cloning site. Now, in vitro, in a test tube, as it contains the COS site, you can package this into a bacteriophage. Now you have these phage particles and you are growing your E. coli. You just infect these bacteriophage to the E. coli, and the bacteriophage is going to transduce the bacteria or the E. coli with the cosmid, which includes your gene of interest. So basically, you are using the bacteriophage to insert the DNA of your interest along with that cosmid into the E. coli. Now, once that transduction is done, it can be maintained in the E. coli, because remember that cosmid also do have an origin of replication for E. coli, selectable marker like an ampicillin, may have LAC-Z, so they can be now maintained into the E. coli. It is just the first step that getting that large DNA into the E. coli is difficult, but once it gets in, which is helped by the phage, it can be maintained. So the function is that you can put a large piece of DNA compared to the normal plasmid by using the cosmids. Now, we are going to talk about even further. So when large fragments of the DNA must be cloned, say for an example, pieces of your chromosome, eukaryotic chromosome, which could be even 1 million base pair, which is 1000 kb. And is it possible? Yes, it is possible as well. Say for an example, let's first take a look at a bacterial artificial chromosome, or BACs. And these are played important role in the Human Genome Project as well. So think about that. This is very, very stable. And this artificial chromosome do have these LAC-Z, a COS site. You have a replication origin site, and these SOP-A sites. These make sure that this artificial chromosome replicate into the daughter cells as well. So this is maintained for generations into the cell as an artificial chromosome. And you have the multiple cloning sites. Right here, you can see that LAC-Z, and these are the names of the different restriction enzymes where you can clone them. Clone the gene of your interest. So what is the advantage of the bacterial artificial chromosome? You can clone almost 300 kb of DNA into this. So 300 kb means 300,000 base pairs. So what is the advantage of that? So what in the I'm just giving a perspective of the Human Genome Project, how it was done. This is called a process like a shotgun cloning. So you isolate the DNA from any cell, like a human cell. You cut it with a restriction enzyme, just one restriction enzyme, say suppose HinD3. So all of your DNA is going to be cut through this HinD3, and see that human genome is 3.2 billion base pairs, 3.23 billion base pairs. So you are going to find a lot number of these HinD3 sites, and it's going to cut the DNA into pieces, like fragments. You cut this bacterial artificial chromosome with a HinD3 as well, whatever, same thing that you are digesting your human DNA isolated. It will open it up. You ligate those human DNA fragments with this. So remember that we saw that in the very first, you have a blue segment, a red segment, and a green segment at the beginning of the chapter. Same way, you will have thousands and thousands of different fragments of different size and shapes. They can be ligated into these bacterial artificial chromosomes. And then what you do, that you isolate them, separate them according to their molecular weights. They could be a little bit different, and then you sequence them randomly. So as you are sequencing them with these, each one of them will have a different fragment of the human genome. You compute them out. Some of them may have overlaps, because you just not only digest with the HinD3 once, you have different samples where you do is APA-L1, BSL1, BAMH1, SPH1. You make these BACs with this shotgun type of cloning. What it means that you randomly cut it with the restriction enzymes, make different fragments, and clone them in the bacterial, in the back or the bacterial artificial chromosome. And now you sequence each one of them, that the different colonies that you're getting. And for each of the restriction enzymes, you are going to get a lot of these overlapping fragments. And you can computationally make out all these overlapping sequences to make the whole chromosome sequence or the genome sequence as such. And it's not as easy as it's being said, but in a very crude way, that's how it helps in sequencing large projects or genome sequencing and cloning as well. On the other hand, the yeast artificial chromosome is even more. Remember that this yeast artificial chromosome is a linear chromosome. BAC is still a circular chromosome. Yeast is eukaryotic chromosome, and it is straight linear chromosome. You have a telomerase at the end of the chromosome. You have a centromere in the chromosome. You have a multiple cloning site. You have a replication site. And then you have an URA3, which is the uracil auxotroph selection as well. So what is the advantage of the yeast artificial chromosome? The advantage of the yeast artificial chromosome is that you can clone almost up to 1,000 kb or 1 million base pair into the yeast. But the yeast artificial chromosome is much less stable compared to a BAC. That's the disadvantage. But these are some of the technologies that is being used in the recombinant DNA technologies these days. So let's take a look at that. What is the polymerase chain reaction? This polymerase chain reaction amplifies the targeted DNA of your interest. And why you want to amplify, that is the main thing. And we are going to look at this, and then we are going to look at that some of this real-time or quantitative PCR to endpoint collection PCR to application for each. And we're going to define those words as well. And summarize the importance of the PCR in biology, and we can explain how the sequences of the 5' end of a oligonucleotide primer become included in the product when they do not have the homologous template. We're going to talk about that and design the oligonucleotide primers, which helps in something which is called a seamless cloning without the restriction enzyme. We're going to talk about that as well. So here, what we are looking at, that the overview of that enables gene amplification. The rapid synthesis of many copies of specific DNA fragments from a complex mixture of DNA. Oligonucleotides are primers or single-stranded DNA molecules, generally between 15 to 30 nucleotides. This is needed. And because the primers are based on the flanking nucleotide sequences, only DNA with known or nearly known sequences can be amplified by a PCR. So let's take a look at how this really works in reality. So before going into the schematics, what a reaction mixture of a PCR reaction will have, it will have a template DNA. Suppose we are working on the human DNA and you have a human template. Then you need 2 primers. These primers you design and commercially make, and these are based on the sequence of your target DNA's flanking sequence. That means suppose you have an ATG to stop codon TAA, TAG, or TGA. This is your DNA. You have to have a flank sequence which is upstream to ATG or start codon, and you have to have a flanking sequence at the 3' end, which is downstream from your stop codon. You are going to design your primers from those regions. That's the important part. So we're going to come back to that again. Then you need a thermostable DNA polymerase, which can withstand high temperature, like a 95 degrees Celsius, 74 degrees Celsius, at least for 3-4 hours. And these DNA polymerases, they are enzymes, and these are proteins. Remember that most of the proteins will denature at 65 degrees Celsius, but these are not denatured and can withstand 95 degrees Celsius for a prolonged period of time. And how this works, these were isolated from specific organisms like Thermus aquaticus. These bacteria and archaea, there are many of them, they grow in hot springs or boiling mud baths, hot water springs like say Yosemite or something like that. And these are often, these extremophiles means that they are basically growing in extreme conditions. And these extremophiles from here, these were isolated, and that was the thing. And this was the PCR was discovered by Kary Mullis, and this completely revolutionized the process that how in 3-4 hours you can make billions of copies of the DNA from one copy. So along with that, you need the 4-deoxyribonucleotide triphosphate, or the DNTPs. These are your nucleotides which needed to be incorporated, or this is needed to synthesize the DNA. This is the basic raw material for the DNA, or 4 of them, ATCGs. You need that. You need a thermocycler, that's the instrument used in this reaction. This thermocycler is a very efficient machine where it can ramp up the temperature very high and it can decrease the temperature very fast. So say for example, 50 degrees to 95 degrees, it can do in 12, 13 seconds. And from 95 degrees Celsius, it can go down to 60 degrees Celsius in again 10, 12 seconds. So it's the ramping up and ramping down is very, very fast. And they have a block. On this block, you can just put your tubes, and the tubes are usually plastic tubes with a very thin wall so that there is good conduction of the heat or the cold. And usually these tubes are like Eppendorf, small Eppendorf tubes, but they are usually smaller, about 0.2 ml or 0.5 ml in size. It's smaller than the 1.5 ml tubes. And you need the double-stranded DNA is denatured first step in the single-stranded DNA. And then you use the primers. You denature the DNA at high temperature, and then you lower the temperature so that these primers, which will find the complementary sequence in the flanking region that we talked about, they will anneal to the template DNA. And then what happens, you increase the temperature to the synthesis temperature a little bit more, and the polymerase, the Taq polymerase or the thermostable DNA polymerase, is going to make the full length copy of each strand. And now you have from one strand, you have 2 strands. And this cycle will go on. You will go back and start back again. So from 2 to 4, 4 to 8, and you are exponentially increasing the number of DNA strands by the amplification process. So how this actually works. Suppose you have your genomic DNA and you have, this is your target sequence. You have produced a primer. So this primer is very complementary to the annealing site, and this annealing site is present, because this is, you have to know, this sequence is present in the flanking region of your gene of your interest from your genomic DNA. So when you put them in the reaction mixture, you just denature them at 95 degrees Celsius. They are going to separate the 2 strands. Hydrogen bonds between ATCGs are going to break, so they are now single-stranded. Now you lower the temperature to about 50, 60 degrees Celsius, depending upon the primer. These primers are going to anneal to the complementary base pairs at each end, like this. This is what is being shown. It's going to do it here, going to do it here as well. Now from the about 50 to 60 degree annealing temperature, you go to the extension temperature, and the DNA polymerase will extend these 2 primers into opposite directions. So this will extend this way, this will extend this way, because remember that it's going to only extend to the 5' to 3' direction, going to extend to the 5', 3' directions. So because see that if you look at that, it is annealing to the 3' to 5' end. This is a 5' to 3'. DNA polymerase is going to extend to the 5' to 3' direction. Now from one strand, you have 2 strands. Now you go back to here again. You again denature is these 2 new primers are going to bind. Now from 2, you get 4. Again, you go back from here, denature, anneal, extend. Denature, anneal, extend. You do about 30 to 35 cycles. And if you calculate the exponential rate, you get 1 billion copies after 30 cycles and of these the same sequence, and which takes about 3 to 4 hours. Within 3-4 hours, you get 1 billion copy of DNA. This process as the endpoint PCR. So you started with a DNA and you end up with a large amount of DNA. And this is used for some specific purposes when you need a large amplification of the DNA to do something with the DNA or to identify something. You need these things. And we're going to talk about that. What are the other processes? You can isolate a particular gene from a complex material like a soil, water, blood. Diagnostics test is huge. Chlamydia, hepatitis, human papillomavirus. Think about that how we do that. Suppose you know we all know the sequences of the hepatitis virus, the whole genome. You just make a primer, which is commercially available, and those primers will have complementary sequence to specific hepatitis virus genes. So if the patient does have suspected that have hepatitis, you take the blood from there, you can just directly put those primers, do the PCR reaction. If there is hepatitis virus in the blood, it will automatically produce a product of your interest. And this is the nice thing about it. Suppose you know that where these primers are going to bind on the hepatitis virus gene. And if you know that what is the number of base pair in between that. Suppose you have a gene which is 1.5 kilobase, 1500 base pair. So you have one end, one primer is binding, another end, another primer is binding. So when the PCR product is going to be produced, it's going to be 1500 base pair long. As soon as your PCR reaction is done, you take the DNA from here, you put it on an electrophoresis gel that we have seen, you run a molecular weight marker. If you see that there is a DNA band at 1500 base pair, you know that that sample is positive for the hepatitis virus infection. If you do not find that 1500 base pair DNA, that means that primer did not find any sample or the DNA into the mixture that has been drawn from the blood, and that's why it is negative. So this is how you can run diagnostic tests. Rapid, sensitive, specific, also employed from forensic science, used in criminal cases as a part of DNA fingerprinting technology. So these are some of the processes you can do. You can just get these DNA fragments, very, very little amount of DNA from different type of samples, from hair, from skin cells, from body fluids or anything like that. And you can amplify that. And after amplification of the DNA, you digest them with a specific restriction enzyme. And when you run them in a gel electrophoresis, it's going to produce a pattern. And this pattern is matched with a database just to see that if that matches with the DNA fingerprint of any particular individual as well. So these are some of the ways that the PCR can be used, and these are all what is called as an end product PCR. We do these all 30-35 reactions, just amplify that, and we just use that. But we talked about that there is something else as well that we are going to talk about, that what is the real time and the quantitative PCR. So your real time or the quantitative PCR is something different. In this endpoint PCR that we talked about, we have no idea that how much DNA we actually started with. The reason for that over the period of time, the amount of DNA that is being produced is not linear. What we mean by that, say for an example, when the DNA polymerase is producing these DNAs, it goes into a curve which is not linear. So what it means that at the amount of DNA it was producing at the second cycle is not similar to the 7th cycle or the 15th cycles or the 18th or the 26th cycle. I'm just saying randomly. So it is not linear. So you cannot tell that how much DNA was in the sample when you started. So at the end, when you quantitate the amount of DNA, it cannot tell you that how much sample you have. Just giving you a sample. You have collected water, say from your local 3 different lakes, Lake 1, Lake 2, Lake 3. And then you are looking for a certain type of gene for a salmonella outbreak or something like that. And you are running a primer with a salmonella specific primer. You can have positive or negative, positive or negative, positive or negative from each of the sample. That is the product of your end product PCR. But you do not have an idea that how much contaminant or how much of the salmonella is in each lake. You do not have any clue. But your quantitative PCR can do that. So the quantitative PCR uses a specific type of block, which does have a small window which can focus a laser onto the tube where the reaction is going. And instead of using just regular deoxyribonucleotides, DNTPs, you are going to use the deoxynucleotides, triphosphates, with a fluorescent label. So what is going to happen that when the DNA is being synthesized, these fluorescent labels are going into the DNA. And the laser scanner can easily quantify the amount of DNA is being produced in real time and make a graph. So you have your lake 1, lake 2, and lake 3, and you isolated very little amount of salmonella from here, good amount of salmonella here, medium amount of salmonella here. So your starting material DNA that you have isolated is different in each. Your real-time PCR or qPCR or quantitative PCR is going to tell you where you have more salmonella, less salmonella, medium salmonella. It's going to give you a curve. So quantification can be done in that quantitative PCR. So that's the advantage of the PCR. Very, very powerful product. Here is another example where you can use a technique which we call as a 5' extension. And by that process, using the PCR, you can make a product or make a gene insert for a vector. So let's see that how this works. So here A and B is entirely separate. So first look at this top panel right here. So here I have a gene of interest. This is a gene. Suppose this is gene X that I want to clone in a vector. Could be any type of vector. So what you have a 5' 3' sequence, a 3' 5' sequence. So you have these. This is your 5' to 3'. This is your 3' to 5'. You have these complementary sequences, double-stranded. Now you design a primer from this region, which is specific to your sequence, gene of sequence, because you know this sequence and it's complementary to this sequence. Now you have this primer a little bit overhang. What we mean by an overhang, your primer also have some extra sequence where you have introduced a restriction site or a sequence which will be recognized by a particular restriction enzyme. So site right here, GAC, GTC. Here, on the other hand, you have introduced another restriction site like AAG, CTT, another restriction site. So when you take your DNA into a PCR reaction, you're going to do a PCR with these overhang, these primers. These primers are not going to completely anneal to the sequence because this overhang sequence is extra that is not there into your sample. It's going to only hybridize or with the matching sequences. So first you denature the two strands, let the primers anneal. They will hybridize in this region, they will anneal or hybridize in this region. You will have this overhang. Now you do the extension. What is going to happen, that remember that how this works, the way it works, that right here you have the 5' to 3' end. So in the first reaction, this is going to extend the DNA all the way to the end. But think about that, what is the complementary DNA strand? You have this right here. This is your 5' to 3'. It's going to extend in this direction. This is your 5' to 3'. It's going to extend all these directions. But just in the next cycle, what is going to happen that this complementary DNA is going to go all the way to the end to fill this gap. And this sequence is now within the total amplified sequence. Now remember that it will just go all the way to the end up to this. It can go all the way to the end up to this. And now you have this full length of these introduced restriction site at one end. You have another restriction site at one end. So at each step, no matter what, it is going to separate the two strands. These primers are still going to bind at this point. After the first round, it's going to find the full complementary sequence, is going to find the full complementary sequence, and is going to just replicate the DNA. And this DNA of your interest will now have the flanking sequence. We call this a flanking sequence with the restriction site of your choice because you have designed this. Now what you can do, that you have your amplified DNA, you cut this PCR product with the restriction enzyme, and you cut your vector with the restriction enzyme. So your vector will have a sticky end. This will have a sticky end. You ligate them. You can ligate that PCR product. So this is the technique of your 5' extension. This is one product. So Daniel Gibson actually taken this to another level, another scientist, which we introduced what is called as a seamless cloning. So here what you do, that you take a vector And suppose you have an area which is green and red, and you want to introduce your DNA in between that red and green sequence. So here you have an insert DNA, and you take that insert DNA as well as your plasmid or vector DNA in two separate reaction mixture, and you digest them with, you PCR amplify first the primer with the vector sequence, and you add the vector sequence. So that's the first thing. So that is very important, because remember that you will have to add this green sequence to this end. You have to add the red sequence in this end. You can easily do that. You have just learned this process right here. So you make a primer which is complementary to your insert DNA, and you just make an overhang of your vector sequence. You make an overhang of this green sequence, and this red and the green sequence is inserted already into your PCR product. Now, here you have your PCR amplified product with your gene of interest in the middle with the flanking vector sequence. And here you have your vector. Now you digest both of them with a nuclease which is known as a T5 nuclease. So what this T5 nuclease does, they chew back these 5' ends. So you will produce a 5 right here, a overhang right here at each end. So you have an overhang right here, 5' 3' end, 5' 3' end, you have an overhang. The same way it will chew up the base pairs back from the 5' end, you have these overhangs on your PCR product. You put them together, these sticky ends will join, and you will have some gaps. So what you do that to this mixture, you add the DNA polymerase 1 and then you add the DNA ligase. DNA polymerase 1 is going to repair these enzymes. Ligase is going to seal that. You have your clone product in between your green and red region. This is your cloned gene. You do not need to have the restriction enzymes used in this process. You just introduce the complementary sites of your vector into the flanking region of your gene of interest, amplify that with a PCR by the 5' extension primer design, and introduce that piece of DNA into your cloning vector. Now let's take a look at that, why you make these genomic DNA libraries and what is the advantage of these. So we already talked a little bit about that. So think about that you have a gene of interest that you have no idea where to find the gene. You have the whole genome and you have to fish it out. So this is where you do the genomic library, used when the gene of interest is on a chromosome that has not been sequenced. And then these libraries are screened for the genes of interest in a variety of ways. And we're going to see that. So a cDNA library can also be constructed for the DNA fragments without the intron sequences. So there are two ways. You can take the genome, digest it with the restriction enzymes or multiple restriction enzymes, which will make these fragments of DNA that you clone into vectors, like large vectors like BAC or the bacterial artificial chromosome. Once you have these fragments cloned into the different bacterial artificial chromosomes, you sequence them and try to find out something. So this is your genomic DNA. On the other hand, what you can do that from the mRNA, you can construct a cDNA library, and you can take these cDNAs and you can insert these cDNAs into a specific type of vectors. So now what you have that in these vectors, each one of them, as you are making the cDNAs out of only mRNAs, this library will have just the gene sequence from start to stop, or maybe a little bit extra of these sequences. So there are two different things. Genomic libraries will have the introns, whereas the cDNA libraries do not have the introns, and different perspective for each one of them. So let's take a see that how we construct a genomic library and what we do. So we take a plasmid and we take the genomic DNA and we do a restriction digestion, which will cut them in the fragments, say A and B. This will be in reality thousands and thousands of fragments. Now we cut the plasmid or the vector by the same restriction enzyme. Now what happens that this will go into this will go into this. So multiple of these plasmids randomly will pick up these and we ligate them and we transform the bacteria. So they will have, each one of them will have these plasmids. And so we will have a mixture of these plasmid, each having a different fragment. So this type of cloning is also known as a shotgun cloning. So in that sense that we are just making these fragments randomly, shotgun, and then we are just adding these fragments randomly into the plasmids and transforming the cells. So each bacteria is getting these different plasmids, and then we transform these. So one bacteria has a red fragment, one bacteria has a B fragment, and then we plate them. And this master plate will have these hundreds and thousands of colonies. We do a replica plating, and with the replica plating, we can isolate the gene of interest is in which fragment. And how do we do this? This is a very simple technique, but there are many other sophisticated techniques that can be used in complicated processes where it is not so easy. Suppose you have a tryptophan here. We are selecting the media against ampicillin, means that this plasmid will have an ampicillin resistance. So only the bacteria with the ampicillin resistant gene will be growing on this media. But this is a complete media. So when we are seeing the complete media, means this has all the different amino acids are present in here. Now we do a replica plating and we put into where we do not have any tryptophan in the media. So here, only the transformants containing the cloned tryptophan biosynthetic gene will grow. Here, if you do not have a bacteria, because this bacteria is auxotroph, transformed tryptophan auxotroph with plasmid. So remember that this bacteria cannot produce tryptophan by itself. Now, we are putting them into a media which allows the bacteria to grow in presence of tryptophan, no problem. We are supplying the tryptophan. So tryptophan auxotrophs will grow here. Then we are replica plating it to a tryptophan negative media. So if the plasmid contains a tryptophan gene, that bacteria will only go into the tryptophan less media. And now you isolate those bacteria and isolate the plasmid, you have your tryptophan gene of your interest. So this is a kind of a scheme that how from this random genomic libraries, which is either the genomic or the cDNA libraries, you can isolate the gene of your interest by a functional assay. There are many different types of assays. This is one of them. Now, the last part that what we are talking about is how we express the foreign genes in the host cells. And what we mean by this. So far, we were talking about that cloning the genes into a vector to produce a large amount of these cloned genes. We were not talking about that the product the gene makes. Now we are talking about the product, which is the expression of the gene, which is going to make the protein of interest out of that gene. Say, for an example, so far we were cloning, say, insulin gene. Now we are going to make that insulin in an organism. Suppose it's a human insulin gene, we are going to make it in yeast or say in bacteria. How we do this and the whole process we are going to discuss. So here what we have done, the clone gene in the new host cells are called heterologous gene and may not be expressed unless they are modified. Because what happens? Suppose the insulin gene, if you just put in the regular plasmid, and the regular plasmid doesn't have any promoter in front of the gene, no RNA polymerase will bind and will express that gene. So what we have, the recombinant gene must have a promoter that the host RNA polymerase recognizes. Means if we are trying to make insulin, human insulin, into E. coli cells, I will have to have a vector, and this vector is called an expression vector. It's not a cloning vector. It's not a cloning vector. The difference in between an expression vector and a cloning vector is that a cloning vector doesn't have a promoter sequence in front of the gene of interest, where an expression vector will have a promoter so that when introduce an inducer into the media, that inducer will allow the expression or production of that protein from that gene. And differences exist in eukaryotic or bacterial host. And mRNA requires a leader sequence and mRNA modification, intron removal. These are some of the things that we have to do. Sometimes even we have to change the codons because bacteria preferentially use a different set of codons, although they are same, but they have a preference over some codons compared to the others. So eukaryotic codon choice and prokaryotic codon choice also varies. And expression vectors are used to overcome these problems with the expression of recombinant gene in the host cell, which are foreign genes, because we are producing the products or the proteins in E. coli from human being, or we are producing these from yeast cells from another human being. So these are all heterologous expression. They are not from the same animals. And we have these inducible promoters, and that result in a high level of transcription. So inducible promoters means that we add a specific chemical into the bacterial growth media, which bind to these promoters and will drive the RNA polymerase to produce a high level of mRNA, and they will be translated by the bacterial ribosomes, organism of interest, whether it's an E. coli or yeast or any animal cell, in a large amount. Remember that this cloning doesn't have to be done in a bacteria or yeast. It can be done into an animal cell as well. Animal cells can produce products as well. Insect cells are used for producing proteins of interest in a large amount as well. That's your expression. So now we have produced, our recombinant cells have produced a large amount of the protein. We can isolate the protein, purify that. Purification is a daunting task usually, and we usually do the chromatographic technique, and we take the proteins through different columns, and small scale in the laboratories, large scale in the industries, to purify the protein and the final product. But most for the purpose in the laboratory and to quickly understand, particularly at the research level, we add a tag to the protein which makes it easier to purify. And one of these tags is a polyhistidine tag, and we're going to talk about that. So this is just one example. There are many different types of tags. That's a flag tag, sumo tag. There are many different commercial names. You can look at that. And all of these actually help. The principle is the same, an easy process by which the protein can be isolated from these thousands of mixtures of these proteins that are present in a bacterial lysate. Also, you can label them in the fluorescent label so that they can be coupled with the green fluorescence or red fluorescent proteins, and they are going to fluoresce under the fluorescence microscope that you can visualize. A lot of different things that we can do. So here we are looking at tagging a protein with a polyhistidine tag. So what is a polyhistidine? Usually it is a 6-histidine that you add either at the 5' end of the gene or at a 3' end of the gene. So think about this, that this is the protein specific. So these are two different things. This is your protein sequence. This is your specific sequence. And here is your start codon. After that, you just put this CAT, CAC. Remember that CAT and CAC, both of them code for histidine. So these are 6 histidine codons that we added at the 5' end toward after the ATG or the start codon. So 6 histidine will be added onto the protein at its N-terminal end when it's translated. Here, the sequence is at the 3' end just before the stop codon. So when the protein is made, this histidines will be added to the C-terminal end of the protein. Most of the times, these tagging of 6 histidines doesn't make the protein non-functional. Sometimes it does. But on the other hand, there are some ways that we can cleave off these histidine sequences by putting a protease digestion sites. So these are all the possibilities that is also possible. So what we are trying to do here. So we introduce these histidine sites, and then we clone this whole thing, either this or this, that's your choice. And this is your whole sequence, you clone it with the histidine tag into the vector. Doesn't matter which end, you can use any end. So then what you do right here, you have, see that how it is, you have your 5' ATG, you have this area, protein specific sequence. So this is basically what you are going to amplify in the process of making the construct. So this is your protein specific sequence, and each end you have your complementary sequences, and you can easily make complementary sequences produce products. So now what you can do, that you clone the gene of your interest with this histidine tag, and then now you're going to purify your protein that is being produced in these cells. So how do you purify this? So you break these cells and make a lysate of this E. coli, which will have the protein of interest produced with the histidine tags. And along with that, you will also have a large amount of your expressed histidine tag protein, along with all the bacterial junk proteins. So what you do, that there is a specific thing that is important. The histidines have an affinity to bind to nickel and cobalt metals. So there is a resin which is just agarose, and these agarose are chemically bonded with something called NiNTA, nitrile acetic acid. So it's a nickel bound to a specific chemical which is tagged to an agarose, which are the beads, and you add this onto a column. So this column is filled up with this specific type of agarose, which do contain a metal at its end. Usually it's called NiNTA. It's either nickel or cobalt. So you fill up this column, and once you just crudely purify your lysate by the initial centrifugation, you take the lysate, which is a supernatant with all your soluble protein, you just pass through them, and you collect the flow through. Then you wash this with a buffer so that all these unbound or non-specifically bound proteins go out, and you collect them as well. And then finally what you do, that add a specific chemical which is known as an imidazole. So the imidazole does have a similar kind of ring structure like a histidine. So it will compete with the histidine to bind to those nickel or metal binding sites. So as you add imidazole, the proteins which were binding by the 6X histidine tags to the metal agarose column or the nickel column, they are going to elute out. So this is the way you purify the protein. Now let's see that we take all these crude flow through, washed flow through, and this is the eluate with the imidazole, and we run this protein gel. So molecular weight marker, then let's see what happens. The first one, this one, is the lysate, which is the flow through. You can appreciate that how much of this protein is right there, because these are all the unbound protein that's passed through the column. See how many of these. Then this is the wash right here. You can appreciate right here that still a lot of protein were bound non-specifically. They are out by the washing. And then when you add the imidazole, you only elute the bound histidine tag protein of your interest. You have the purified protein in this fraction. It is usually not that clear that easily, but still it is pretty good for this. So this is how you purify a tagged protein. So the other ways that you can do the labeling of the protein to add a red or green fluorescence protein with your gene of your interest. So you can add these green or red fluorescence protein. You can have many different types. And the tagging of these protein means that you make a chimeric molecule. You have a protein of your interest, and to the N-terminal or the C-terminal site, you add this 27 kilodalton protein. Usually it's about 25 to 30 kilodalton protein, and your protein is going to express as a fusion protein. Sometimes it will lose the function, sometimes it will not lose its function, and you can look at the localization of your protein of your interest into the organism if you introduce that protein where it is going. So you can easily look at the fluorescence microscopy and look at these with the tagged proteins. So this is your chimeric fluorescent protein labeling by producing a chimeric molecule where you fuse a green, red, or any colored fluorescent protein with the protein of your interest to identify where they are in the cell. So this last part that we are going to talk about is the Cas9 nuclease. This is a precise tool for the genome editing, and these were initially discovered in the bacteria, where they fight the bacteriophage infection by destroying the bacteriophage DNA. But now this can be used in the eukaryotic system as well. This is also known as a CRISPR or CRISPR-Cas9. So Cas9 is a nuclease that acts in the bacteria used to find some of these sequences. So these are clusters, regularly interspaced short palindromic repeats. So again, palindromic repeats means they forward, backward, they read the same way. And why this CRISPR-Cas9 system was in the bacteria, as I said, that this is the way that they found some of the residual sequences of the bacteriophage. The overview of the story in just a few words, that over the period of time when the bacteriophage infect the bacteria, some of the bacteria were able to survive. And at that time, that bacteria will take some of the sequences of the bacteriophage and incorporate them into the CRISPR closer to this Cas9. So what will happen that this CRISPR sequences will be propagated into the bacterial population, which are actually the sequences from the viruses or the bacteriophages, which are the viruses for the bacteria. When there will be an attack of these bacterial viruses or the bacteriophages, the CRISPR-Cas9 can find these sequences and they can chop these sequences out. So this is actually used widely for altering genomes in vivo. So you can just use this system with the CRISPR-Cas9, which you use a guide RNA with the Cas9 nuclease. You can just introduce them into the cell, or you can clone these Cas9 and just introduce cells or the animal which is expressing Cas9. You can introduce the guide RNA to that specific cell or the tissues, and you can do a genome editing or disruptions. So again, lots of different modifications of this can be done. So let's take a look at how this works. So here you have a Cas9 nuclease, and the Cas9 nuclease will only cut a piece of DNA if that is bound to a specific RNA, which is called a guide RNA. So how this guide RNA is known? The guide RNA will have a specific sequence that needed to be bound to the Cas9. This is a fixed guide RNA sequence. This is the Cas9 nuclease recognition sequence of the guide RNA. So that means that the Cas9 only understands this sequence, that this is the guide RNA. A random sequence of single-stranded RNA or mRNA will not be recognized by the Cas9. When you have the genomic DNA, suppose you want a specific segment of the genomic DNA you want to disrupt, or you want to replace it with the sequence of your interest. So what you do that, if you want to disrupt, you make about 20 base pair complementary sequence of your DNA, and you have the specific guide-specific RNA sequence on the other end. You have the Cas9 nuclease either introduced or expressed into the cells or the tissues, and you introduce this RNA into those particular cells. So what's going to happen that, as you know, that when a DNA during the transcription process is going to separate out, and that at that time, when the DNA is during the replication process or any other process, when the two strands are going to get separated, these complementary RNAs are going to bind to these DNAs, which is the complementary sequence of the DNA RNA. So if you have a DNA RNA hybrid, this will bring the Cas9. So wherever this guide RNA will bind, Cas9 will go in there and chop that sequence off. So what is the consequence of that? Suppose you have this end chopped off, and at that time, if the two ends join, like here and here, if the two end joins, and this is called a non-homologous joining. So if a non-homologous joining happens, if you just randomly take out some base pairs out of that, probably if it is inside a gene that you want to silence, the whole sequence of that gene could be completely frame shifted. And the product that you want out of the gene is completely going to be, it's not going to form because you are silencing the gene. So this is how you can disrupt a gene by having a sequence specific to the gene and introduce a drastic frame shift mutation. So you silence the gene. On the other hand, you can modify a gene or edit that gene as well. So here, what you have to do, that you have a specific sequence the same way, But along with that, you introduce something else. You have to use a donor DNA, which will have a sequence in the middle, that of your choice, but it will have a sequence from the flanking sequence of the guide RNA. Again, repeating, this is the sequence you choose for your guide RNA. And here you have a flanking sequence, a 5' flanking sequence, you have a 3' flanking sequence, and you introduce a donor DNA, and this is the piece of donor DNA that you want to introduce where this cut will be. You want to introduce this piece here. In the donor DNA, you should have a sequence from here as well as here to the target sequence. So what will happen that you have the guide RNA, you have the Cas9 nuclease, it will chop it off at the same way, but at the same time, here you will have this donor DNA with the similar sequence of this flanking sequence. And there could be a homologous recombination because these sequences are the similar, these sequences are the similar, so the homologous recombination can join a complete new molecule. Here, see that this is a non-homologous recombination, and most of the purposes, this is a disruptive, this is for gene disruption. Whereas this one, you are introducing a new sequence, and this is for editing purposes. So you can edit by the homologous recombination, whereas you can disrupt by a non-homologous recombination. And this is the power of gene editing by the Cas9 nuclease using the guide RNAs. So this is one of the technologies that recombinant DNA technology is going to. So that's basically the overall tool that we talked about. Some of these techniques are, well, there could be some of the gray areas because if you do not work with these material with your hand, sometimes it's difficult to correlate. And if you have any questions, please post them in the discussion thread or try to use with those search words, and that may actually help you to find some of the answers from the YouTube. There are many resources out there to try to find them in there. But you can always post these questions and I can always find it for you or answer them as I can. That will be the end of chapter 17, and that will be the last material for your module 2.